Morning wishes to all my dear students. Students, welcome back to the science class. I hope you all must have enjoyed your Diwali vacation. Now, let's get started with our second unit test portion. Students, we are going to start with lesson number 10, cell and cell organelles. Okay, this is a new lesson for you. It belongs to biology discipline of science. Okay, so let us start with the topic cell and cell organelles. Chapter number 10, Cell and Cell Organelles. Students, we have been introduced to what is cell and what are the different types of cell, that is animal cell and plant cell, in standard 7th. We have already learned what is cell all about. Yes. So let us have a quick revision, okay, about the structure of animal cell and plant cell for you. Okay, now before that, how many types of cells are found in living organisms? How many different students? There are different types of cells which are present in living organisms depending upon the organs, yes, and the tissues. We have different types of cells. Okay, which instrument had you used to observe the cells? Okay, now one instrument definitely I would talk about is microscope. Electron microscope is used to observe cells in detail. Okay, students, animal cell and plant cell, these two basic types of cells we already know. Let us have a quick revision about all the organelles, yes, or the components of the cell. Yes, so this is a diagram for you all. We will start from top to bottom. First we have rough endoplasmic reticulum, then we have smooth endoplasmic reticulum at the bottom. Yes, rough and smooth. We have a nuclear envelope, nucleolus and nucleus. These are all the components of the cell. Okay, that is at the center. Nucleus is at the center, right? Then we have a plasma membrane, which is like a boundary or a covering of the cell. Lysosome, mitochondria, ribosomes, Golgi body, centriole. These were all the parts of the animal cell. Okay, so I hope this is revised now. To define cell, students, cell is the structural and functional unit of living organisms. We have already learned how we have atom as a basic unit of matter. Similarly, for organs or tissues or the organ system, cell is the structural and functional unit of it. Okay, so now according to the functions, what happens is cells, they have different sizes and they have different structures, okay, which are found in different organs. As I said, different organs have different functions, so depending upon that, the cell will be also different. Okay, coming to plant cell, if we revise, we have almost similar structures are there, like we have rough endoplasmic reticulum, smooth endoplasmic reticulum, we have nucleus at the center, which is a center point, then we have chloroplast that was absent in animal cell, lysosome, vacuole. If you see, vacuoles are large in size, okay, they are much bigger than animal cell. Then cell wall, Golgi complex, cytoplasm, cell membrane, ribosome. Okay, so these are all the components of a plant cell. Okay, these things we have already drawn, revised in standard 7. Okay, so I hope this is clear. Now in this chapter we are going to study in detail about the cell organelles also, what all things are present inside the cell and what are its functions and all about. Okay, so I hope the introduction of the chapter is all clear to you all. Later concepts we will see in the next video. Thank you. Welcome back to the science class, students. Students, in the previous video we have revised the definition of what is cell. Okay, two types of cell we have revised, that is plant cell and animal cell, in detail. Okay, so let's move ahead to the further concepts of this chapter. Okay, cell and cell organelles. So are you all ready? We are learning chapter number 10, cell and cell organelles. Students, so we are going to learn more about cells as I promised. Okay, in this chapter we are going to understand all the components of a cell and its functions in detail. Overall, yeah, and or overview of the components of the cell, if you closely watch or you closely observe, it is divided into 4 subparts. Okay, if I want to talk about components of cell, I am going to talk about the 4 different parts of a cell first. First, we have cell wall. Second, we have plasma membrane or cell membrane. Third, we have cytoplasm. Fourth, we have cell organelles. So students, we are going to learn all these 4 subparts in detail in this chapter. Okay, so let's get started. The very first part, that is cell wall. So what is cell wall all about? Students, cell wall is found around cells of algae, fungi and plants. I repeat, it is found around cells of algae, fungi and plants. That means what? That means animal cells completely lack the cell wall. Animal cells do not have cell wall. Okay. Now, cell wall is an elastic and strong coat around the cell membrane. Okay, how it is? What are the features of the cell wall? If you see, it is elastic. It has a strong coat around the cell membrane. Cell membrane, plasma membrane is same. So around the cell membrane or plasma membrane, what do we have, students? We have cell wall, and that is only and only present in algae, fungi and plants. Now it is mainly composed of what? It is mainly composed of carbohydrates. Now what are the types of carbohydrates which it has? It has cellulose and pectin. Please remember the words cellulose and pectin. Afterwards, polymers like lignin, suberin, cutin, they are added in the cell wall as per the requirement or as per the need. Now students, what is a polymer? Polymer is a useful chemical which is made up of repetitive units. Okay, we have examples of polymers like lignin, suberin and cutin which are added along with carbohydrates. Okay, so in overall, if you see cellulose, pectin, lignin, suberin, cutin, these are all made up of, yes, or these are all the components of the cell wall. Okay, we can say like that. Now what are the functions of the cell wall? Very, very important, students. Cell wall is going to act like a support. Okay, it is going to act as a support and it is going to protect the cell by preventing entry of excess water in the cell. Okay, so it is going to act as a barrier, or it is going to prevent the entry of excess water inside the cell components. Okay, so let us see the structure of cell wall. With this is a very much same figure which is present in the textbook. Okay, if you see cell wall in detail, we have plasma membrane, and above the plasma membrane, can you see the cell wall? Primary cell wall. We have certain soluble proteins present inside. We have cellulose microfiber. As I said, it is made up of carbohydrates. So cellulose, pectin, hemicellulose, middle lamellae, these are all the parts of the cell wall. Okay, cell wall is present after the plasma membrane or the cell membrane, and present only and only in algae, fungi or fungi and plants. Animal cells do not have cell wall at all. Remember this. Now let us see plasma membrane or cell membrane. This is the second part, second subpart. Students, plasma membrane and cell membrane, okay, it is present beneath the cell wall, down the cell wall. Yes. Now if animal cells doesn't have cell wall, that means animal cells directly has plasma membrane or cell membrane as the outer covering or outer coating. Correct? Now let us understand this in detail. It is a thin, fragile and elastic covering that separates the cell components from other environment. Okay, it is going to act like a covering and it is going to separate the cell components from other outside environment. Clear? Now let us understand the structure of plasma membrane. Students, please go through the figure. Protein molecules are embedded into layers of phospholipids. Can you see lipid layer? These are nothing but phospholipids. Okay, 2 layers of phospholipids are present, and if you see, there are proteins which are embedded. Can you see the carbohydrate chains in blue color? So protein pink color they have shown, and we have carbohydrate chain in blue color. Okay, so this is the structure of plasma membrane. Very simple structure it is. Now, very important property or feature of plasma membrane we are going to understand. Plasma membrane is said to be a selectively permeable membrane. Now, what is selectively permeable membrane? Student, it is going to allow some substances to get an entry and it will prevent some substances, okay, other substances to go out. Okay, so it is going to allow some substances to enter the cell and it will prevent some other substances. Okay, so selectively permeable. Selectively permeable. Okay, now this particular property of the plasma membrane, it is very much useful. Okay, if you see useful molecules of water, salt and oxygen, they can enter the cell. Now which useful molecules? We have water, salt and oxygen, they can enter the cell, and carbon dioxide can exit the cell, which is a waste product which is not required. So that will exit the cell. So certain things will entry, yes, certain things will exit. So this is selectively permeable. Now if any changes occur outside the cell, the cellular environment does not change. The cellular environment means the components inside the cell. Okay, it is very much compact, it is retained, and the environment is not changed due to plasma membrane. And this condition is known as homeostasis. This condition is called as homeostasis. Okay, I hope the selective permeable membrane word is clear to you all now. We have already seen cell wall structure and plasma membrane structure. The further concepts of this chapter, okay, about cytoplasm, about organelles, about different functions, we are going to see in the next videos. Till then, thank you and take care. Very good morning to all my sunshines. Students, we have already started with second unit test portion. Yes, we have started with lesson number 10, cell and cell organelles. In that we have seen what is cell, different types of cell, that is animal cell and plant cell, correct? Then we started with components of a cell. Yes, in that we have subparts, 4 subparts, remember? So in that already we have seen what is cell wall, what is plasma membrane and cell membrane, in detail its structure. So in this video, a new concept related to plasma membrane, cell membrane and cell wall we are going to study. So let's get started. We're learning chapter number 10, cell and cell organelles. Students, we have already discussed that plasma membrane or cell membrane is a selectively permeable membrane. It is known as a selectively permeable membrane, where it will allow the entry of some substances and some substances can exit from the cell. Remember carbon dioxide, which is a waste product which is given out of the cell. Now a very important question arises here is, how do substances travel in the cell? This question must be wondering, you must be wondering about this concept, that how the substances they come inside and outside the cell. So we are going to study that only in this video. Basically students, if you see, there are certain activities, there are certain processes that the cell carries out. Okay, now there are certain activities which are carried out with the help of cellular energy. And there are certain processes where you do not require any cellular energy to take place. The cell doesn't require any cellular energy to carry out that particular process. So the very first part, that is activities that consume cell energy, in that we have two types or two processes, endocytosis and exocytosis. Second is the process which don't consume cellular energy. In that again we have two types, diffusion and osmosis. Okay, so let us see all these processes in detail. Okay, activities that consume cellular energy. Okay, in that we will see the very first definition of endocytosis. Okay, what is endocytosis? Endo is inside. Okay, something taken inside. So to gulp or engulf food and other substances from outer environment. Okay, anything which is coming inside the cell is endocytosis. It can be food, it can be any other substances. Second, we have exocytosis. Now what is exo? Out. To excrete, to give out. So to excrete waste materials out of the cell. Clear with the two definitions, endocytosis and exocytosis. And these two processes, they will require cellular energy. Okay, now let us see the second part, that is processes those that don't require or consume cellular energy. Okay, in that very first type we have diffusion. Now what is diffusion? Simple definition we have as entry or exit of small molecules like oxygen and carbon dioxide. Very few small molecules, they can take an entry and they can exit. In and out, they can do this process. That is diffusion. Second type we have osmosis. Very, very important, students. Okay, the definition is very important. Its types, the whole concept is very interesting. Okay. Now see, the traveling of water from a part with more water to a part with less water through a selectively permeable membrane is osmosis. Okay. Now what is happening here? Water is traveling from where to where? From a part with more water, okay, with high concentration, to a part with less water or less concentration. Through what? Through plasma membrane or cell membrane, which is a selectively permeable membrane. Remember, we have already learned this. Okay, so this is osmosis. Now students, osmosis is a physical process and there are three possibilities related to it. Now depending upon the three possibilities, we have three types of solution: isotonic solution, hypotonic solution, and hypertonic solution. So let us see all these three types of solution in detail one by one. What is isotonic solution? Medium outside and inside the cell has same proportion of water. Medium outside the cell and medium inside both has same amount of water, proportion of water. So what will happen? Water doesn't go in also or out also. There is no movement or no traveling of water. That is known as isotonic solution. Let us see hypotonic solution. What is hypo? Hypo is less. So less water where? Inside the cell than the outside medium. So the cell has less water than the outside medium. So water enters the cell. Water will enter the cell and this is called as endosmosis. Entry of water inside the cell. Very good example, students, we have of resins, that is manuka. Okay, when you keep in water, after some time what will happen? These manukas which were very shrinked, okay, now they have swollen up and they have become turgid now. Okay, so that is a very good example of hypotonic solution. Hypertonic solution, students, can you guess now what will be definition of hypertonic? The cell will has more water inside than the outside medium, and in this case what will happen? Water will travel outside, it will come out of the cell. Okay, it will just come out of the cell. Very good example we have is fruit pieces when it is kept in a very thick saturated solution, sugar solution. Okay, so outer medium we have less water and inside, if you take fruit pieces, like example of watermelon, we have lot of water. Yes, water concentration in watermelon is more. What will happen? Watermelon will shrink and all water is given out of this, out into the sugar solution. Okay, this is a very good example. So if the fruit pieces they shrink after a while, this is the actually observation after you put the fruit pieces in the sugar solution. Now if plant cell or animal cell is kept in hypertonic solution, what will happen? Water will come out of the cell and this coming out of the cell is known as coming out of water. Okay, process is known as exosmosis and there is contraction of cytoplasm, and this whole process of exosmosis and everything where we have contraction of cytoplasm is called as plasmolysis. What it is called as, students? Plasmolysis. Let us see the figure. Now you all know what is hyper, iso and hypotonic solutions. Can you see the very first figure, osmosis in plant cell where water is coming out? That means it is hypertonic solution. Second is isotonic solution and third is hypotonic solution where water is getting inside the cell. Similarly, if you see, compare it with the animal cell, it is the same thing. Okay, oxo water given out, same amount of water, no movement of traveling of water. Then we have last, we have entrance of or entering of water molecules inside the cell. So this was the osmosis in plant cell and animal cell figure. I hope this is clear to you all now and it was very interesting, right? Okay, the next concept we will see in the next video. Till then, thank you. Welcome back to the science class, students. Students, in the previous video, that is video number 1, we have understood the concept, how substances travel inside the cell, different processes like we have endocytosis, exocytosis, diffusion, osmosis, okay, all different types of solutions we have learned, right? Now let us move to the third sub part of components of a cell, that is, let us understand cytoplasm in detail. Very, very important component of a cell. Okay, so let's get started. We are learning chapter number 10, cell and cell organelles. Okay, so the very third subtype of components of a cell we are going to learn is cytoplasm. Students, let us see it in point wise. Okay, let us understand it in point wise. Cytoplasm is the fluid between plasma membrane and nucleus. Now, plasma membrane, it is present outside. It is the outer covering, right, of the cell. And where is nucleus? Nucleus is at the center. So at the center we have nucleus and plasma membrane. So the space between plasma membrane and nucleus is filled with some moving sticky substance which is called as cytoplasm. Okay, I hope you can imagine what I'm talking all about. Now we all know that many cell organelles are present inside the cell. So all these cell organelles are what? They are all suspended in the cytoplasm. It is not air or vacuum or something like that. We have a sticky substance which is called a cytoplasm, which is very much present, and in that all the cell organelles are suspended. Next point is cytoplasm is the medium for cellular chemical reactions. Students, lot of activities, lot of processes are carried out inside the cell. So it is going to serve as a medium, okay, for all the cellular reactions. The part of the cytoplasm other than the organelles is the cytosol. Yes, the part of the cytoplasm other than the organelles, okay, apart from organelles, it is a cytosol. Cytosol stores some vital substances like we have amino acids, glucose, vitamins, etc. What are amino acids? These are the building blocks of proteins. Basically, when you break down a protein, what you will get? You will get amino acids. How we have matter, and then when you divide the very smallest part of a matter, okay, that is known as atom. Similarly, when you break down proteins, you get different types of amino acids. In animal cells, cytoplasm is more granular. Many granules are present in animal cells and it is very dense, while in plant cells it is thin and mostly pushed to the periphery. Why? Because we have large central vacuoles in plant cell. Remember, in plant cells we have large vacuoles. So because of this large vacuoles, what happens? Cytoplasm is pushed to the periphery. Pushed to the periphery means to the boundary. Okay, so cytoplasm is more granular and dense in animal cells as compared to plant cell. Very, very important points. So I think you will be able to write 4 points, any 4 points of cytoplasm here. Let us understand the this figure. This is nothing but your onion peeling. Okay, onion peeling. Okay, these are the cells which are observed when you can take the inside peeling of onion. If you take, okay, if you stain with some solution, so you will get such kind of cells can be observed under microscope. So if you see this rectangular shapes, whatever is seen is nothing but cells of onion peeling. The thick structures, can you see? Okay, pipeline like structures, this is nothing but the cell walls. We have a proper dark circular structure which is known as nucleus. Then small granules, can you see? Dot structures we have, that is cytoplasm. And then we have a space, lot of space, that is known as vacuole, okay, which is present very much at the center, that is vacuole. So this is a plant cell. So this is onion peeling as an example of plant cell. So I hope this figure is clear to you all. So that was all about cytoplasm. Further concepts we are going to see in the next videos. Okay, thank you and take care till then. Good morning wishes to one and all. Welcome to the science class. Students, in the previous video we have seen the third subtype of component of the cell, that is cytoplasm. We have understood all the different features, different properties of cytoplasm. Okay, we have seen this figure, the structure in detail of onion peeling also. Students, now in this video, let us start from this video. Basically, we are going to start the fourth sub part of component of cell, that is cell organelles. Different types of cell organelles we are going to study one by one. Okay, in that the very first type or the first part, important center of the cell, which is known as nucleus, we are going to study it in detail. So let us start together. We are learning chapter number 10, cell and cell organelles. Okay, the very fourth okay, subtype of components of the cell, that is cell organelles. Let us understand basically what are cell organelles. Students, an organelle, okay, is a specialized subunit having specific function within the cell. Now all the cell organelles, these are nothing but these are specialized subunit. They have their own specific functions inside the cell, and they are known as organs of the cell. How in human body we have different organs, okay, and all these organs have different functions for us. Similarly, in a cell we have cell organelles which are having definite or different functions, we can say, of a cell. Each organelle has its own lipoprotein membrane. Every organelle has a membrane, that is lipoprotein membrane. Students, except nucleus and nucleus and chloroplast, all the cell organelles, it can be seen only with the electron microscope. Okay, if I want to see the nucleus and chloroplast, I can see it in the compound microscope, but in detail, if I have to see other organelles of the cell, I have to use only and only electron microscope. So this is a picture of electron microscope which is given in the textbook. Okay, so this is the electron microscope. Okay, so let us start with nucleus. Okay, in this video I'll be only talking about nucleus. Students, this is the center point, or which is the controlling center of the cell, I must say. Nucleus is all about different metabolic activities which are coordinated and supported, is everything carried out with the help of nucleus only. So let us understand nucleus with the help of an activity. What I have to do, we have to take a drop of water on a clean glass slide. I hope you all know after science experiment videos what is a glass slide. You have to use an ice cream spoon, and you have to gently scrape the inner surface of your cheek. That means this is something, the material or the sample will be from the inner surface of the cheek, okay, inside. With a needle, transfer a little material from spoon to the water drop on the slide. Now whatever, after scraping whatever cells you will get on the spoon, that you have to take a little, you have to take on the slide, and then you have to spread it evenly. We have to stain this, okay, since it is colorless, I want to stain it with methylene blue solution on the smear, and then cover slip will be added and we can observe it under microscope. So this is a simple activity that we can do. So whatever blue nucleus I'm going to see, okay, whatever blue colored structure I'm going to see, it is nothing but nucleus, okay. That is the main motive behind doing this activity. Students, the dark round spot which is seen under the microscope while observing the onion peeling stained with iodine is the nucleus of the cell. Remember, while studying the cytoplasm, that onion peeling structure we have seen, in that can you see the nucleus? We have already discussed about nucleus, so that is stained with iodine solution, okay. Okay, this is the structure of nucleus, okay, when observed under electron microscope. Now circular structure is seen, but in detail if I want to study, I have to observe it under electron microscope. So we can see nucleus is covered by a double layered membrane. It is a double layer membrane, and there are small pores or holes which are known as nuclear pores. These pores allow the passage of material in and out of the cell. Okay, they have a specific function. It will allow the material to come inside and the materials to go outside the nucleus. So that is the function. This is the function of the nuclear pores. Nucleus has one round nucleolus. Can you see this purple colored structure? That is nucleolus, and a network of chromatin fibers. Can you see lot of chromatin fibers? It is like a mesh like structure or interconnected structures. That is nothing but chromatin fibers. So nucleolus, chromatin fibers, nucleopores, nuclear membrane. These are basic structure. These are the basic structure of a nucleus. Chromatin fibers are thin thread-like structures which condense to form short, thick chromosomes. Chromatin fibers, students, it is going to form chromosome at the time of cell division. Okay, when the cell, when the process of cell division arrives, at that time, what happens is chromatin fibers, they are going to condense, they are going to shrink into chromosomes. Functional segments on the chromosomes are called as genes. So the very much famous term genes has arrived now. Okay, so where are genes present? They are present inside the nucleus, and these are functional segments of the chromosome. Okay, so genes are present as functional segments on the chromosomes. What are the functions of the nucleus? What are the main functions of the nucleus? 2 points I will discuss here. First of all, it controls, I said it is a controlling center, main center, controls all the metabolic activities of the cell and also the cell division. So metabolic activities can be like formation and breaking down of some substances, some important chemicals, okay, which are very much important for the cell. Everything is controlled by the nucleus. Second is it is involved in the transmission of hereditary characters from parents to offsprings. Hereditary characters like in the form of genes. You must have heard that you look like similar to your mother or father, your parents, right? Now these everything is expressed, or it is a gene expression, okay, from parents to offsprings. So who carries out this transmission? Nucleus, because nucleus contains genes. Okay, so I hope this both functions are clear to you all now, and nucleus was very interesting, right? The controlling center. Next organelles we will see in the next upcoming videos. Till then, thank you. Welcome back to the science class, students. Students, in the previous video, that is video 1, we have seen the first cell organelle, nucleus. Remember, we have seen that. Okay, now in this video I'm going to talk about the second cell organelle, that is endoplasmic reticulum, ER, endoplasmic reticulum. So let us see that in detail. We're learning chapter number 10, cell and cell organelles. Okay, so the second cell organelle is endoplasmic reticulum. Before that, students, just answer me a few questions like how many types of pipelines are there in our building or home? Water pipelines, we have gas pipelines, and we have drainage pipelines also, okay, for the disposal of sewage. So there are different types of pipelines. Then their functions are really different functions we have. So what will happen, okay, if these pipelines don't exist, or if they are not there, or if they are not present? Okay, so they have some functions, right? Very important functions. Okay, so let us begin with the explanation of endoplasmic reticulum. This is the figure which is given in the textbook. Very interesting. We are going to represent, or we are going to relate endoplasmic reticulum as pipelines. Okay, now the organelle which conducts various substances inside the cell is called as endoplasmic reticulum, which conducts various substances where inside the cell is called as endoplasmic reticulum. So ER, which is a short form of endoplasmic reticulum, it is nothing but what? It is like a net. Okay, it is net-like structure consisting of interconnected miniature tubes and sheets filled with fluid. Can you see? They are all interconnected with each other, the tubes and sheets, and they are all filled with a particular fluid. Inner side, if you see endoplasmic reticulum, inner side, it is connected to the nucleus, and the outer side is connected to the plasma membrane. Plasma membrane is the outer structure, right? Nucleus is the center, at the center. So this is like a pipeline, this is like a channel. Students, endoplasmic reticulum, they are of two types basically. We have rough endoplasmic reticulum, we have smooth endoplasmic reticulum. You don't have in detail about that, but please remember that this rough endoplasmic reticulum, okay, it has got ribosome granules. Can you see ribosome structures are shown? These are present from the outer surface. So we have ribosome granules on the outer surface. So that is the structure of endoplasmic reticulum. Now, what are functions of endoplasmic reticulum? It is like a framework. Okay, it is a framework that will support the cell. Conduction of proteins. Okay, they are going to help in the conduction of proteins. Toxins that have entered the body through food, air and water are made water soluble by endoplasmic reticulum and then flushed out of the body. Whatever toxins that have entered the body through any medium like food, air and water, it is made water soluble by ER and then it is flushed out of the body. So very important functions: framework, conduction of proteins and removal of toxins from the body. I hope endoplasmic reticulum is clear to you all now. Okay, we have two types, RER and SER. Okay, further cell organelles we will see in the next video. Till then, thank you. A very good morning to all my sunshines. Students, how's everything? I hope going great. Okay, well, students, we have started with lesson number 10, cell and cell organelles. In that, we have already covered components of cell subparts. That is, the first thing we saw was cell wall. Then we saw plasma membrane or cell membrane. Next, we saw what is a cytoplasm. Then we started with the fourth subpart, that is cell organelles. Now, under cell organelles, if you can recollect, we have completed nucleus. That is the very most important cell organelle. Nucleus we have completed. Then we have also seen what is an endoplasmic reticulum. We have seen the structure, the description, explanation we have seen, and the functions of it is also covered. Okay. Now let's start with the next cell organelle, that is Golgi complex. So are you all excited to learn this new cell organelle present in the cell? Okay, so let's begin. We are learning chapter number 10, cell and cell organelles. Students, we are going to see and study Golgi complex in detail. Okay, if you see the structure, that is figure 10.9 Golgi complex we given in the textbook. Okay, after going through this figure or the diagram, let us see the explanation one by one. Point number 1 says Golgi complex is made up of 5 to 8 hollow and flat sacs placed parallel to each other. Hollow and flat sacs. Yes, can you see how parallelly they are arranged? Cisternae. Okay, the name is cisternae. Okay, these sacs are called as cisternae. And students, the cisternae are filled with different enzymes. Okay, in earlier videos also, I remember I have just given the definition of what are enzymes. Enzymes are nothing but proteins. These are substances which speed up the chemical reaction. Okay, they assist in the chemical reaction, but they don't participate in it. They don't participate in the reaction, but they will assist the reaction. Okay, that is the definition of enzymes. What are enzymes? These are nothing but proteins. Okay, so Golgi complex made up of 5 to 8 hollow flat sacs placed parallel to each other, and these sacs are nothing but cisternae, and they are always filled with enzymes. The proteins which are coming from endoplasmic reticulum. What is ER, endoplasmic reticulum? Students, we have seen the function of endoplasmic reticulum in that the second function was conduction of proteins. Okay, transfer of proteins. Okay, from one part to another. Now, the proteins which are coming from endoplasmic reticulum, okay, they are enclosed in vesicles. Okay, if you see the figure, fusing vesicles words are written. See, fusing vesicles, some circular structure is there. Okay, these vesicles, okay, they are coming from endoplasmic reticulum. They will come towards Golgi complex via cytoplasm. Okay, so the proteins coming from ER, they are headed towards Golgi complex via cytoplasm. Cytoplasm will bring this vesicles, and these vesicles contains what? Proteins. Okay, next what happens? These fusing vesicles, as the name suggests, fusing, so they are going to fuse with something. So they are going to fuse with the formation phase of the Golgi membranes. Okay, now the formation phase is at the above, okay, at the top. So can you see cis phase? Now there is a place, or that is a place where these vesicles will fuse with the Golgi membranes and empty their contents. Whatever they carry inside, proteins, substances, that will enter into the inside the cisternae. So they will empty their contents in the cisternae. So after fusing, all the contents in the vesicles are given inside the cisternae of the Golgi complex. Now as they pass through the cisternae, they are chemically modified with the help of enzymes. They are again packed in the vesicles. What happens is, after fusion occurs, okay, after fusion with the formation phase of the cisternae, okay, now they are modified, okay, the structure is modified with the help of the enzymes which are present in the cisternae, and then again they are packed in the vesicles. So we have fusing vesicles, and down can you see new vesicle, secretory vesicle. So these vesicle circular structures which are coming from downside, okay, can you see these two structures? They are nothing but new formed vesicles. Okay, now these vesicles, they will exit from the maturation phase. So maturation phase is at the bottom, formation phase is at the top. I hope it is making clear. The idea is very clear now. Thus, cisternae work like a packing department. So it's like a packing department. Fusing vesicles, they will fuse with the cisternae, then they will be modified, and then again they will be packed into new vesicles, and these new vesicles are given out from where? Maturation phase of the cisternae. Okay, so packing department this is. Now let us understand functions of Golgi complex. Students, very important function is, it is a secretory organ of the cell. Secretory substances are secreted, as I said, proteins are modified inside it, and again, okay, new enzymes are secreted. It modifies, sorts and packs material synthesized in the cell. Okay, for example, we have enzymes, mucus, proteins, pigments, etc., and dispatches them to various targets like plasma membrane, lysosome, etc. So accepting it, modifying it, sorting it, packing it, so many things are done, right, by Golgi complex. Very interesting. Third is, it produces vacuoles. Students, we all know that vacuoles are present in animal cells and plant cell both, right? Plant cell is in much bigger size, we have vacuoles. And secretory vesicles, as we have seen, okay, you know that secretory vesicles are released or secreted from the maturation phase. Fourth, it helps in the formation of cell wall, plasma membrane, and lysosomes. So they are very much involved in the formation process or synthesis process of cell wall, plasma membrane, and lysosomes. Okay, so read the functions of Golgi complex again carefully. Okay, introduction to scientist. Students, Camilo Golgi, he described the Golgi complex for the first time. So that is why you must be wondering why the name is Golgi complex. So let's remember the scientist Camilo Golgi, who has described the Golgi complex for the first time. He developed the staining technique which is called as black reaction, and with the help of it he studied nervous system. So these are his certain contributions. He won the Nobel Prize of 1906 along with Santiago Cajal for the study of structure of nervous system. So he has great contributions in the field, okay, of studying different systems. That is one of the very important system, that is nervous system. Okay, so this was Camilo Golgi. Students, I hope you have understood Golgi complex, why the name is Golgi. Okay, what is the structure of the Golgi complex and what are the functions of it. Okay, later organelles or next organelles we will see in the next video. Thank you. Welcome back to the science class, students. Students, in the previous video we have seen what is Golgi complex all about. Its structure, its function, everything was made clear, right? I hope you have understood that. Now let's move to the next cell organelle, that is lysosomes. I repeat, the next cell organelle is lysosomes. Okay, so let's begin. We are learning chapter number 10, cell and cell organelles. Students, in this video we are going to learn what are lysosomes. Okay, before that let me ask you one question. Okay, students, what happens to the agricultural waste after a few days when it is dumped into a compost pit? What happens to the agricultural waste? Yes, the answer to this question is probably that most of the bacteria and fungi, they act upon this complex waste product, break down into simple substances, and then that is utilized by the soil. Okay, that is mixed with the soil and then utilized by the plants. Okay, so that may be the probable answer for this. Okay, so that is very much related to what are lysosomes in the cells. Okay, let us see that. Students, due to various metabolic activities in the cell. What is metabolic activities? As I said, what is metabolism all about? Anabolism and catabolism, that is formation, that is synthesis and breakdown. Okay, that was given as science article homework. The definitions are already given to revise. Okay, again I will try to explain what are metabolic activities. There are certain activities which are carried out inside the body. Okay, which is very important. Okay, synthesis of some larger structures are important. Similarly, breakdown of larger substances into simple substances, that is also important for a cell. Okay, for our body. So what happens, due to these various metabolic activities? Organic waste is generated. Okay, organic waste is generated. Now what to do with this waste? Who are going to help us out? Lysosomes. Lysosomes, they will digest the waste. They will break down the waste. Okay, and how they are going to carry out? Students, lysosomes are very simple in structure. If you see on my right hand side 10.10 figure which is given in the textbook, you can see an oval shaped structure with a single membrane and enzyme complex, or different enzymes are present inside this particular structure. So it is simple single membrane bound sac. Okay, and these sac are filled with different digestive enzymes. Okay, enzymes which are very much involved in the digestion process of the waste. Okay, so filled with digestive enzymes. Very simple. Okay, functions of lysosomes, let us see one by one. This is very important. Okay, the very first function of lysosomes is they are related to, okay, or they are a part of our immune system. Now what are they going to do for us? Students, lysosomes destroys the viruses and bacteria that attack the cell. Okay, when our body, okay, is attacked by different viruses and bacteria, they destroy these viruses and bacteria. They take an entry inside the cell. Okay. Second most important function of lysosomes, demolition squads. Okay, so let us understand this. What is demolition squads? Okay, what is demolish? Is to destroy, right? So it destroys worn out cellular organelles. Okay, and organic debris. So this particular process of destroying the worn out cellular organelles and organic debris is known as autolysis. Auto is self, means it is involved in the self-destruction activity also of the cell. Okay, which are worn out, cellular organelles which are worn out, that is digested by the lysosomes. So that is why they are known as demolition squads. Okay, clearing out, cleaning process. Third, we have suicide bags. Yes, suicide bags, you heard it right. When a cell becomes old or is damaged, okay, either the cell has become old now or is damaged due to any injury or any other accidental cause, lysosomes, they burst. I mean, they open up. And what happens when they open up? Enzymes which are present inside, the digestive enzymes which are present inside the lysosomes, they come out. And what these enzymes will do? They will digest their own cells only. Own similar cells are digested by the lysosomes. Okay, so that is self-destruction kind of thing. So these are nothing but self-destroying is suicide. Yeah, so suicide bags, you can correlate that way. Fourth, we have very interesting point is during starvation. Okay, when you don't have food for a longer period of time, lysosomes, they digest the stored proteins and fats in our body. Okay, so during starvation, what happens? Lysosomes digest, store proteins and fats in our body. So these were the functions of lysosomes that we have seen one by one. Okay, I hope it is clear to you all. So we have seen Golgi complex, first video. Second, we have seen lysosomes. Next video, let us understand what is mitochondria. Okay, so thank you till then. Welcome back to the science class, students. Students, in previous two videos, we have seen what is Golgi complex, what are lysosomes, their functions, their structures, everything is covered, right? So in this video, let's understand mitochondria, a new cell organelle for you. Let us understand structure of mitochondria, functions of mitochondria, everything we will learn in this video. So are you all ready and excited? Okay, so let's start. Students, we are learning chapter number 10, cell and cell organelles. In that, we are going to understand mitochondria. Before that, a small question to all of you. Okay, which type of energy is required to run the fans, computers and electric bulbs? Okay, which are very much present in our homes, our houses. How do we run them? Yes, the answer is with the help of electrical energy. Electricity is important to run these devices, right? Okay, so let us understand mitochondria in relation to that. Okay, students, each cell requires energy, and this energy is produced in the mitochondria. So you guessed it right. That means mitochondria is related to energy, for energy production. Yes, it is very much related. So let us understand that. Under the electron microscope, if you see the structure of mitochondria, okay, under the electron microscope, a mitochondria is seen as a double membrane structure. Okay, double membrane. Double membrane means we will have a inner membrane and we will have outer membrane. So it is double membrane structure. The outer membrane is porous. Porous means it will have small holes on it. And the inner membrane is deeply folded. Can you see how it is folded? Yes, overlapped and it is folded. Okay. So energy production. Okay, double membrane structure. And we have inner membrane, outer membrane. Okay. So that is seen. Now let us understand about the folds. Okay, now these folds that I talked about, okay, which is very much present inside as inner membrane, are called as cristae. The inner cavity is filled with proteinaceous gel-like matrix containing ribosomes, phosphate granules and DNA. DNA is what? Deoxyribonucleic acid. We all know what is DNA. Genetic material. Yes. So inner cavity is filled with ribosomes, phosphate granules and DNA. Students, let me here tell you, or let me here share you a small piece of information. Okay, that in detail you are going to learn in standard 9th and 10th. Okay, proteins production or protein formation. For protein synthesis or protein formation, students, we require certain materials. Okay, we want certain organelles here. Which organelles? Ribosomes, we want phosphate some granules and DNA. So when these three things are present as materials, we can produce proteins. So here, since mitochondria contains all these materials or cell organelles, we can, or mitochondria can produce its own proteins. Okay, so that was the next point. Now, with the help of enzymes, mitochondria oxidize carbohydrates and fats in the cell. Dear students, mitochondria has the capacity to combine carbohydrates and fats with oxygen. What is oxidize? That is ability to combine with oxygen. Yes. So with the help of enzymes, mitochondria can readily combine carbohydrates and fats with oxygen. That is the meaning of the sentence. the energy released in this process is stored in the mitochondria in the form of ATP. Okay, so during the process of oxidation of carbohydrates and fats, okay, energy is released. And where it is stored? It is stored in the mitochondria. In which form? In the form of ATP. What is the full form of ATP? Adenosine triphosphate. I repeat, what is ATP? Adenosine triphosphate. Okay. Students, plants cells have less mitochondria as compared to animal cells. And the reason behind is very obvious. Plants don't do physical activity. Can plants move? No. Animal moves. Animal, human beings, we creatures, we move from one place to another. We have lot of physical activity to carry out. And for that, we want lot of energy. That is why lot of mitochondria is present in animal cells as compared to plant cells. This is the simple reason behind proportion of mitochondria, why it is less in plant cells and more in animal cells. I hope it is clear. What are the functions of mitochondria? Students, as I said, energy production. So what does mitochondria do? It produces energy rich compound. Which compound? ATP. As I said, they oxidize carbohydrates and fats in the cell, and during this process energy is released and it is stored in the mitochondria in the form of ATP. Second, we have synthesis. What is the meaning of synthesis? Simple formation of. Okay, so what is synthesis? Synthesis of proteins, carbohydrates, lipids, etc. Lipids are related to fats. Okay, they are a version of fats only. So proteins, carbohydrates, lipids, etc. by using the energy in ATP. Okay, in higher classes you will learn more in detail how energy is utilized or used, because we have ATP triphosphate. We have 3 phosphate molecules here, bonds here. And when these bonds are broken, energy is released. So that complete process you have in 10th standard. Right now, what you have to remember that, okay, mitochondria are majorly involved in energy production. Powerhouse of the cell. You can call it as powerhouse of the cell, right? So I hope you are clear with mitochondria. Okay, next class we will see the next cell organelles. Till then, thank you and take care, all of you. Good morning, standard 8th class. Students, in lesson number 10, we all are learning different types of cell organelles, right? We have already covered cell organelles like nucleus, yes, endoplasmic reticulum, Golgi complex, lysosomes, mitochondria. Students, let's move to our next cell organelle, that is vacuoles and plastids. Okay, so let us understand vacuoles and plastids. So are you all ready? Okay. We are learning chapter number 10, cell and cell organelles. Students, we are going to understand what are vacuoles. We all know that vacuoles are present in animal cell and plant cell both. In plant cell, they are in much larger, okay, they are much bigger size as compared to animal cells, right? They are much larger in plant cells. Okay, so vacuoles are the storage sacs for solid or liquid contents. They don't have any typical shape or size. The structure of the vacuole changes according to the need of the cell. Very, very important. They don't have any typical shape or size, students, but the structure of the vacuole, it changes according to the need of the cell. Depending upon the need, depending upon the requirement of the cell, they change their shape. Okay, vacuole is bound by single membrane, so it is a simple structure. Vacuoles are having a simple structure. They are like storage bags. Remember these keywords. What are the functions of vacuoles? Let us understand what are the functions of vacuoles. The very first important function of vacuoles are to maintain the osmotic pressure of the cell. Students, to maintain the pressure of the cell, let us understand this particular function in detail. We all know what is osmosis all about. Yes, it is a movement of water from an area of low concentration of the solute to an area of higher concentration of the solute. Here solute means we can take as water. Osmotic pressure is what now? We know what is osmosis. Now what is osmotic pressure? Osmotic pressure is a pressure which is created by water moving across a membrane due to osmosis. Okay, so the more water moving across the membrane, the higher is the osmotic pressure. A very good example of osmotic pressure, students, we can take here is filtration of water. Okay, so that is the meaning of osmotic pressure. Second function of vacuoles we have is to store metabolic byproducts and end products. Very, very important. Metabolic byproducts means after metabolism, whatever products are formed, those are known as byproducts and end products, right? Now, these examples can be like glycogen, okay, proteins, water, etc. Now, glycogen is a new term for you, students. Let us understand what is glycogen, okay, at the end, okay, after completing all the 4 points, I will explain what is glycogen. Okay, third point is in animal cell, they store waste products and food, while in amoeba, it stores food before digestion. So, in animal cell, they store what? These are storage sacs now. So, they store waste products and food. Okay. Fourth point, in plant cell, vacuoles are full of cell sap. Cell sap is a liquid, okay. Cell sap, and provide turgidity, rigidity to them. So turgidity meaning we all know that it is like swollen kind of structure is turgid. Turgidity is swollen. Rigidity will be like inflexible. So depending upon, okay, the concentration of the liquid in the vacuoles in plant cell, okay, they can either provide turgidity or rigidity to the plant cells. Okay, that is the meaning of the fourth point. Okay, now I promised you to talk about glycogen. What is glycogen, students? See, our body breaks down the most carbohydrates. Okay, whatever carbohydrates we have, it is broken down into glucose. We all know that. Now, when the body doesn't need to use the glucose for energy, okay, when the body doesn't need to use the glucose for energy, it stores it where? In the liver and muscles. It stores it. Now, this stored form of glucose molecules, which are like, they are connected one after another, is known as glycogen. These stored glucose molecules are known as glycogen. I hope I have made my point clear. Next cell organelle, let us understand plastids. Okay. Students, one question to all of you. Why are plant leaves green and flowers red, yellow, orange or blue? Yes, have you ever wondered? We say plants are green, but if you carefully observe the different parts of the plant, they are either red, yellow, orange or blue. Okay, so the answer behind this is because of an organelle which is called as plastid. The plastid gives such colors to the plants and it is present only in plant cells. Remember, plastids are always present in plant cells. Okay, now plastids, they have double membrane and they are of two types. Okay, double membrane and are of two types. Now, which are those two types? Leucoplast. Leuco meaning is white. Remember, leuco means white, or you can also call it as colorless. So white or colorless plastids are known as leucoplastids. Then we have chromoplast. Chromoplast is colored plastids. Chromo means color. Okay, so remember these two types. Chloroplast can get converted into other types of chromoplast. Chloroplast, which is in green color, which is a green color, okay, that can be converted into other types of chromoplast, that is other types of colors. A very good example is raw green tomatoes. Now raw green tomatoes are green in color, obviously. Now they are going to turn red. Why? After some time, after some duration of time, they become red? Because there is breakdown of chlorophyll and synthesis of lycopene. Lycopene is red in color. It is a carotenoid and it is red in color. So the moment raw green tomatoes, when they turn to red tomatoes, remember one thing, chlorophyll Okay, there is a breakdown of chlorophyll and there is synthesis of lycopene, which is a chromoplast. So this is a table which is given. We have so many colors of the plant, like we have green color as leaves. If I'm talking about carrot, it is orange. Yes, then the leaves, when they become yellow, purple, blue, these are the different color plant part colors, and we have respective pigments which are present in those color. Okay, in those colored plant parts, like if it is green, then what is present? Chlorophyll. If I am saying orange, carotene is present. Yellow, xanthophyll. Purple or blue, it is anthocyanin. For dark pink, it is betalain. Students, this is not the end. We have so many different colored plant parts and we can have different pigments for it. Okay, so I hope you are clear with vacuoles and plastids, both of the cell organelles. Next video, I'm going to talk about chloroplast. Till then, thank you so much. Welcome back to the science class, students. Students, in the previous video, we saw what are vacuoles, yes, and understood plastids and its types, right? In this video, let us understand chloroplast and its function, okay? Its structure, everything we are going to see in detail in this video. So let's begin. We are learning chapter number 10, cell and cell organelles. Okay, students, let us understand chloroplast. Okay, chloroplasts are important for the photosynthesis process that takes place in the leaves. We all know that leaves contain chlorophyll, yes. So chloroplast, they are present in the leaves, yes. They are very much important for the photosynthesis process. We all know what is photosynthesis. We have already learned in standard 7th. Okay, now let us understand what this chloroplast or chlorophyll does for us. Chlorophyll in chloroplast traps the solar energy and converts it into chemical energy. It is going to trap the solar energy from the sun, which is available at free cost for all of us, yes, and convert it into chemical energy. Stroma in the chloroplast. Now, if you go through the figure which is given, 10.13 chloroplast figure, if you see, you have so many different structural components. If you see, we have thylakoids, stroma, granum, inner membrane, outer membrane, and intermembranous space. Okay, so thylakoids are like stack of circular shapes which are placed one upon another, and stroma, it is the internal structure, and then we have granum. Okay, so stroma in the chloroplast contains what? Students, they contain enzymes, DNA, ribosomes, and carbohydrates. So these all materials they contain, that is, okay, and these materials are necessary for photosynthesis to carry out. So these substances are present where? In the stroma. Stroma contains all the substances which are very much important for photosynthesis process to carry out. Okay, let us understand functions of chloroplast. The very first function is very obvious, that chloroplasts convert solar energy to chemical energy. Chemical energy in the form of food. Yes, they store food. Okay. Second point, chromoplasts give different colors to flowers and fruits. Yes, chromo means color. So these chromoplasts are going to give different colors to flowers and fruits. Then we have leucoplast. Now what leucoplasts are going to do? Leucoplasts are involved in the synthesis. Synthesis means formation and storage of food like starch, oils, and proteins. So leucoplasts are involved in synthesis process, synthesis and storage. Chromoplasts, they are involved in giving different colors to flowers and fruits. So now here starch word, let us understand what is starch, students. Starch is a type of carbohydrate. What is starch? Starch is a type of carbohydrate, but it is a complex carbohydrate. Okay, remember this. And this complex carbohydrate, it is made up of long chain of sugar molecules, that is glucose molecules. Okay, now some starchy foods that we have, okay, that we consume are like peas, corn, potatoes, beans, pasta, rice and grains. They contain a lot of starch in them. So these were a few examples to make you understand what is starch. Okay, very, very important point, students. Mitochondria and plastids. If you can recollect, when we were studying mitochondria, I said mitochondria contain all the materials which are required for protein synthesis. Yes, so here mitochondria and plastids both can produce proteins on their own, as well as they can replicate themselves. Replicate themselves means they can make their own copies. Yes, they can make their own identical copies. Why? How is it possible? Because they contain their own DNA and ribosomes. Okay, DNA and ribosomes are very important materials, students, to produce proteins or to have replication process to work. Okay, so this is the important point. I hope chloroplast is clear to all of you now. Okay, its structure and functions. In the last video, we are going to compare eukaryotic cell and prokaryotic cell. Okay, till then, thank you. Welcome back to the science class, students. Students, we have reached to the last section of this chapter. Okay, so far we have understood in a cell the different cell organelles, yes, in detail, their functions, their structure, yes, everything we have covered one by one. Okay, now it's time to compare prokaryotic cell and eukaryotic cell in detail. In standard 7th, we already have learned prokaryotic cell. Now, since we have learned eukaryotic cell in detail, let us compare both of them one by one. Okay, so let's start. We're learning chapter number 10, cell and cell organelles. Students, after studying the cell and cell organelles, you all must have come to know that all the functions in plant and animal cells are taking place smoothly in harmony because of cell organelles, which are very much well defined, yes, specific, and they have their own function to carry out in the cell. Very distinct cell organelles we have learned, students, and such developed cells are called as eukaryotic cells. Yes, as I said, in standard 7 we have already learned prokaryotic cells. So let us compare both types of cells in detail. So let's start. Comparison between eukaryotic cell and prokaryotic cell. Eukaryotic cell and prokaryotic cell, point wise, let us discuss and compare. Okay, let us first discuss about size. Okay, size of the cell of eukaryotic cell is 5 to 100 micrometer. For prokaryotic cell is 1 to 10 micrometer. Clear? Second, we have number of chromosomes. Okay, number of chromosomes is more than one for eukaryotic cell. Okay, if you can recollect what were chromosomes, remember chromatin, they condense at the time of cell division to form chromosomes. While studying nucleus in detail, we have learned about this. Okay, so number of chromosomes more than one. In prokaryotic cell, it is only one. Okay, so let us see the third point. Nucleus is with nuclear membrane, nucleolus and nucleoplasm. So well defined structure we have for nucleus, okay, in eukaryotic cell. As compared to prokaryotic cell, we have a structure called as nucleoid, and that is very much resembling to nucleus. So they don't have nucleus, but they have nucleoid. Okay, functions will be similar like nucleus in prokaryotic cell. Coming to fourth point, mitochondria and plastids are present. Okay, for prokaryotic cell, they don't have membrane bound cell organelles. So we have learned that every cell organelle has a membrane. It may be single layered, double layered, but they have membranes, right? So in prokaryotic cell, we don't have any membrane bound cell organelles. Let us see examples for eukaryotic cell. Present in highly evolved unicellular and multicellular plants and animals. See, multicellular plants and animals, they are eukaryotic cells only. But if you see the higher versions or the evolved form of unicellular organisms, okay, they can have these all features of eukaryotic cell. Coming to prokaryotic cell, very good example is bacteria. Bacteria are prokaryotic in nature. Okay, these are very good example of prokaryotic cell. I hope the comparison between eukaryotic cell and prokaryotic cell is well defined. Yes, to you all now. Okay, with this, I complete the lesson number 10. Immediately, you will get a PDF of question answers. Please write the question answers in Science A4 size long book. Okay, give a nice heading as second unit test and start writing question answers for it. Okay. I hope this lesson is clear to you all now. Okay, next lesson we will start in the next upcoming days. Okay, thank you so much.